Good afternoon. This is week 2 of 4, and it's kind of a mishmash, as this whole subject is, actually, as it turns out. Good afternoon, and it is. I didn't expect to see half of you here on such a beautiful day. Seems inappropriate to be talking about aging on such a beautiful day, but anyway, there are, as we've seen in the leaves, autumn is coming, so we're reminded of those beginnings, interim and endings, that cycle of life, and so we're seeing it in the weather, and you'll be reminded about it in this lecture. Donna Hatton had an interesting idea, and that was to ask you if you have comments to make, not only on last week, but this week, or questions that might not have been addressed, that you might want to kind of write them out on a card. And she has little interesting pens to go along with them. So did you want to hand them out now?
Sure.
Yeah, sure. It's a good idea. And I promise to pay attention to some of them. Depending on whether I can read them. Yeah.
Try to say that legibly.
Now, let's just, it's time to kind of move along. There are 4 essays that I've written. One for the late report on aging. It says everything I'm saying in 4 weeks in one piece. So if you want the summary, it's there in 2 pages. You don't really have to be here. This is a beautiful one on immortality. So it's one I wrote, but immortality, Oliver Sacks and me. We do kind of agree on some things. It's a wonderful one. One on mini brains. So that's the subject of the last one, or part of the subject of the last one. Funerary practices. Well, we've all been to the undertaker and seen some of their practices. Well, we're going to talk about some of that today, but I actually wrote an article on that. So that's included in the little packet from the library. And there's this wonderful poem. Billy Collins is one of my favorite poets, and he wrote one about the afterlife. And what we imagine the afterlife might be. What are the possibilities for the afterlife? So I'm not going to ruin it by telling you about it or reading it out to you. So it'll be in the packet. That itself is worth kind of looking at. So, on we go. Well, just this is a little off. It seems a little off, but it really isn't. This is Einstein, as you unruly hair. He was at Oxford in 1932-33. And, Hitler was actually in government at that time, and he could see the writing on the wall, and he was getting out, as many physicists and other scientists were doing, getting out of Germany. And he was the most famous physicist, maybe the most famous scientist of his day. So it was quite a coup for Oxford then, and still is, to claim him, kind of partly. He was there for only 2 years, but one of the things I'm pointing out here, 33, he was incredibly productive very early in his life, in his career. In 1 year, when he was 22, he wrote 4 papers, any one of which would have garnered a Nobel Prize, for sure. One did. And only 22, working in a patent office, not even in a university. No link to a university. Incredible. And then there's this long period after that, before he wrote his masterpiece, what I consider his masterpiece, General Relativity, in 1915-16. And then really, other than the debates with various physicists about, quantum mechanics and the nature of quantum mechanics, the Nobel Prize was actually awarded a couple of years ago over that controversy, and but not productive. So this gets into the age-related productivity, and certainly physicists, theoretical physicists, and he was certainly that, the primary example of it. His best work was very early in his career. And general relativity, his masterpiece, he was, what, I think I've written it at the time, 37 when he completed it. Took him 10 years to do that. 10 years, with a lot of help from other mathematicians who knew more about math than he did. And he turned down Oxford. He was there for 2 years. You know what a very congenial person, as I understand, certainly in that period. But he didn't like the stuffiness and the ceremonial part of Oxford. He wanted quiet. He wanted to be by himself. That's why he moved to Princeton. And because it was kind of off the beaten path, certainly in that period in the 30s. So I put that up because it's an interesting picture historically. He's obviously earned an honorary degree here from his outfit. But it's a wonderful example of productivity. And here's another wonderful example. One of my favorite magazines is the New Yorker. And not all the articles, probably one in an issue that kind of turns my crank, maybe less than that. This one got my attention. I mean, this lady, the author, I mean, the writing in the New Yorker is very good. You want to learn to write? Read the New Yorker. But it's about a young artist, deliberately put in young, the writer. An Italian widow is still discovering the joy of painting at 93. It's a wonderful story. She lost the love of her life a few years before. She's a very outgoing, social person. Rebecca, I think, kind of fell in love with her. I mean, she was there for a lot of social occasions, and Isabella was a very good host. She lived in a kind of a communal building. Her studio was on the first floor, and her apartment on I think the 6th floor. And it's interesting, I mean, Italians knew about her art, but it's only just in the last 2 or 3 years that the world has found out about her art. And now she has great exhibitions planned in Paris and in New York and in the UK. I mean, she's a big star now. What I like about this picture, I love it. I mean, she may look a little frumpy, but you know, she's got character. You blow up this picture, she has a lovely face. And look, she's standing in front of one of her pieces of art. There's another one kind of dangling over there, and another one. She's a very, very creative artist, and it shows you, it gets into this question, is there an age for art? And I know that some of my artist friends claim that artists can be very productive late in life, and that there isn't this cognitive falling off. I can't comment on that because I don't know enough about it, but I do know about science and the people who do that and the ages when they're most productive, and the Nobel Prize series reinforces that every year, and this is the 8th year. Productivity is in that late first third of life to the kind of the first half of the middle third of life. That's when most people do their really best work in science. Not necessarily when they're best known, and certainly not when they get a Nobel Prize, which may follow 40 years later. You have to survive long enough. But anyway, isn't it a lovely picture? I just love that picture. It's a great one about a person. Well, this is something that you may have noticed. I mean, do I follow bit columns? No, but because of this aging series, I've been paying attention to a bit column. So when people write about the dead, and who do they write about? Well, you know, if it's a high-profile paper, it's going to be an important person. And they're probably going to spend most of the bit telling what the person did to earn their fame. And but have you noticed sometimes that some people drop off the map? So people who are famous entertainers or whatever. And if somebody asks you, well, what's happened to Frank Sinatra? What happened to so-and-so? And maybe go, oh, I don't know, I don't see. There's kind of a blank several years, and then there's the obit. There obviously something happened medically in those last few years. That's why they're out of sight and, in a sense, out of mind. And there's another thing about genetic legacies here. If you think that there's a sense of immortality in your genes, forget it. Because every one of you here is half father, half mother. And in the next generation, it's a quarter, then an eighth, a 16th. So if you go out, what is it, 200 years, 10 generations, just 10 generations, your contribution to that 10th generation is 1 in 1,000. And the next, just add another one, and it's 1 in 2,000 plus. So this is the nonsense of these hereditary shows focusing on things, because they purport to suggest, you know, that these influences passing through time. That's crazy. That's not what's happening genetically. It just isn't happening. So it took only 200 years, 10 generations. Now, that's assuming a generation is 20 years. Imagine with hunter-gatherers or early humans, as soon as the first period was there, or the first sexual urges, people were probably having kids as early as possible, and they didn't live long. I mean, the max might be 30 years. But these days, it might be 30, right? Because women are having children later, and fewer of them, and families later. But for the purposes of this, 20 years. So it's an interesting thought. Well, long-living animals and trees. Well, I was just up in the Bruce Peninsula, and it's very, very different on the Lake Huron side of the lake and Georgia Bay side, because on the Lake Huron side, the trees are stunted. I mean, they hardly grow. They might be up to here and be, you know, 200 years old, or even 300 years old. And just stunted. And you see this on the east coast of Nova Scotia, where the ocean, the influence of the ocean, the harsh weather, whereas my son is on kind of an interior lake in Nova Scotia, and it looks like a Tom Thompson painting. And that's only 15, 20 miles inland. Makes quite a difference. So on the Georgia Bay side, it looks like Tom Thompson. On the other side, they're all stunted, the trees, and kind of makes you wonder about this idea of dietary restriction. You live longer. These trees aren't consuming as much energy over time, or the energy isn't there in their environment, and they live a long time. They're living longer than the trees on the Georgia Bay side. So, well, the others, isn't this impressive? Bristlecone pine, over 5,000 years. That means the tree rings will report the history of the last 5,000 years plus. Isn't that wonderful? Yeah, so that's a repository. How long well, this is an interesting question. Is there a limit to how long humans can live? I mean, there are very, very few, 100 or more. We're going to talk about that in a few minutes. Is that actually a limit, a physiological limit, biological limit? Can the limit be changed? Apparently not, at least in the last half century or century. Most of the increases come because of changes in public health. The obvious ones, sanitation, with the Trump group, not like this one, vaccines. How many of you watched it last night? Fess up. Oh, so half, a little more than half, okay. I didn't. I didn't want to mess it up for Harris. So artificial fertilizer, sterilization of water, milk, I mean, all of that stuff's pretty obvious. But you know, this isn't a sexy subject, public health, but it had a huge impact in late 1800s and 1900s, this century we lived in, a good chunk of our time, looking at most of this. Well, I already said that. Oh, here we are. Well, here are examples of the oldest documented modern day humans. Well, one is Jeanne Coleman, 122. She died in 1997. She reported or said that she met Van Gogh and didn't like him. He was ugly, ungracious, impolite, and sick. And that was her take on Van Gogh. She also ate 2 pounds of chocolate a week and smoked right up to the end. So, amazing. The one reported in the New York Times, the obit column, Ellie Watkins, so what was it, August 23rd this year, died at 117, the oldest living person at that time. But those numbers are, you'd think with the population going up to, I think it's already at 8 billion now, well ahead of what was thought it might be, that those numbers would be kind of creeping up, but they're not, which makes me wonder if there is a biological limit to how long we can live. And then there's a whole question of, I mean, most people would like to live very well and then go boom, right? I think that's what most of us would like. That's my goal. Now, you've seen this kind of thing. I keep bringing out stuff, but I can tell you, nature has reinvented this slide at least 3 or 4 times since it came out. It's one of the most important illustrations in all of science in the last 2 years, because it showed, number one, the spectacular thing is that we might not be here. We came, pre-humans, came very close to total extinction over 900,000 years ago. Now this is based on modern genetic studies. The total population for pre-human, any pre-hominin or pre-human hominin, the total estimated world population was around 100,000. We're not talking towns, cities, or villages here. We're talking about a lot of small groups scattered. And probably family-sized or extended family-sized or a few family-sized thing. Very similar to hunter-gatherers. And then what happens, what happened, bingo, 900,000 years ago, look at that. Literally hanging by a genetic thread. Because the estimated number dropped from around 100,000 to less than 2,000. And the stunning thing is, it remained that way for over 100,000 years. So imagine, you don't have many breeding pairs here. Genetically, it's not a good thing. It's a recipe for extinction. That's what we worry about with rhinos and elephants. The number gets too small for a healthy genetic mix. And surely, pre-humans came really close for a long time. And even then, when they rebounded, not too much, just a little over 20,000, until around the period when modern humans appeared, around 100 to 200,000 years ago. That's when the exodus within Africa and out of Africa took place. So it may be related to the evolution and appearance on the scene of modern humans. It may have been. But the interesting thing, it isn't just a genetic study. If you go looking for fossils in that period, they're really hard to find. So it isn't just made up stuff from genetic studies. The number of fossils became rare at the same time. And there was a huge genetic change. This is where, I'm not going to trip on anything around here, but there was a big change, big shake-up in the chromosomes, because two chromosomes kind of joined up, 2A and B, to become one chromosome 2 as it is now. So something big happened in the genome. We don't know what kind of did it, but nothing else like of that scale has happened since, around the period of this extinction. And that chromosome 2, the combined one, is home to the largest protein encoding gene. Can you imagine the 280,000 base pairs? That's a huge gene. And it produced a huge protein. So a lot was happening, and people are going back to this data and mining it for more and more information. But what's fascinating, we would not be in this room and I would not be talking if that thread had gone pop at some point, and there would be no modern humans, Neanderthals or Denisovans, or, to put them in the framework, Neanderthals appeared on the scene around here almost 400,000 years ago, and their cousins in Asia, the Denisovans, but modern humans down here, anatomically modern humans around 200,000 years ago, behaviorally modern humans around 100,000 years ago. Or roughly when the Great Expansion took place in population and the dispersals. Isn't that interesting how stuff begins to fit together? It's like a giant puzzle on a huge scale. And it's hard for me to think about my own life as having much significance looking at a scale like that, on a grand scale. We don't know what any of these people were like, or they didn't leave much behind, or if anything, artifacts, stuff on walls. I mean, the cave art is really 50 to 100,000 years ago, roughly the time of modern humans and later Neanderthals. Okay, so we should put our own importance in this in its proper scale, I think, which is, it really is an important perspective, I think. Well, and I've mentioned this before, there have been evolutionary thresholds. Now, these often overlap, I mean, and how would you actually date these? But certainly language, symbolic language, and imagination. Imagination, underline that 10 or 20 times. And this awareness of future death. Animals don't have that. They're aware of death, or the imminent death, but as far as we know, maybe not so aware of that they might die in a year or two. But children are aware of death. You know, something happened to grandma, granddad, or something in the family, some with their teacher, something, and they're aware of that. And then this threshold of reverence for the dead. You know, for 99.999% of hominin history, if you drop dead, that's where you decomposed. You stayed there, things ate you, and you chemically decomposed, biologically and chemically decomposed. But to pay special attention and prepare the dead in some reverential way, in a special place, protected from carnivores, that says something about the mind, what they're thinking of, I think. And then the awareness of an afterlife. You can't possibly have an awareness of this without that. Imagination. That sense of there's something beyond this time. And you're not going to get any answers from science because we don't have tools for that kind of stuff. And probably linked to that, an awareness of supernatural forces, spirits, and beings. This is so true of almost every culture that's been looked at that I have to say it's innate to humans, and perhaps Neanderthals. We don't know enough about them, but I think enough. Yeah, probably we do. And I'm reminded of this because I was listening to a homily from the brothers in Boston the other day about All Souls Day, and you know, I haven't paid much attention to stuff like this for years, but it's really quite moving because it's this sense, this almost haunting sense of a connection with those people who have been important to you in your life. And most people want to believe that there is some connection. We hope that way. There's no way of proving that. And oddly enough, in Jan and in her much diminished state in the late stage of Alzheimer's disease, we sometimes have conversations on this very matter. Sounds odd, doesn't it? But we do. And so there is some. It's ubiquitous. Some people claim it's you know, it's a product of the brain, and of course it is a product of the brain. Ultimately, it is. Where else is the imagination? Imagine all of this stuff. But is there something beyond that? I'm not here preaching to you about anything. I'm just saying that this is a very common thing, and we are now talking about the end of life and beyond it. And realizing that there is this cycle throughout the universe, isn't there? The beginning of the universe, the Big Bang, the creation of first stars. They each have a lifetime, an interim. They burn out, often explode, seed, the formation of new stars and that kind of thing, to all life forms. You know, we may be sitting in a hole, but we're eaten up by other life forms, and that continues on. And the atoms go on. Not so much the molecules, but the atoms go on. Anyway, back to this. Some animals are aware of that. Remember the story, I certainly remember in Boston, the cat Oscar in the chronic care home, where I think a lot of patients had malignancies. And the cat had an uncanny sense for who was in the last hours and would jump up on the bed, not in an intrusive way. Mind you, if we were in that bed, we might be saying, shoo, shoo. Yeah, so, and the creation mythologies, bodily resurrection, the Abrahamic traditions, if you're a Hindu or a Buddhist, Billy Collins is quite funny about these. Resurrection of the soul, and what is the concrete parallel for all of that? Elaborate burial ceremonies. And how far back do they go? And of course, in the modern era, even if you don't believe in all of that stuff, you certainly want your name going on. So you might want your name on a new university or a new department or invested in a chair, and your name is on the chair. Or you see this on PBS News every night, right? With all the donors.
Yes.
There, and so people want their name going on beyond them. There are not many anonymous donors, as far as I know. I'm not criticizing, I'm just saying that's the way it is. And well, how far back do we go? Well, a long time, as it turns out. Going back to that slide of the tiny little thread, the early hominin, pre-human, 900,000 years ago. Well, around 800,000 years ago, there is evidence of some care being taken, some reverence for the dead in the choice of where they're laid and protected. And then this peculiar species discovered by Lee Berger and a group in South Africa, very hard to date precisely for other reasons we won't talk about now, and a species with a brain about the size of a chimpanzee, but with some very modern features to the shape of the brain and to the limbs and the hands and the feet. And remember I showed a picture by Rembrandt, or two of the three ages of Rembrandt? Well, you know, I think about this. I've met John Gurgey a couple of times, and he creates these things from fossils. And I think, you know, Rembrandt he's really, really good. But so is John Gurgey. He's not human, so we're not going to hang him on a wall, or most of us wouldn't. We only hang people who look like us on walls. But it took them a long time. So there it is with a kind of forehead, some kind of like us, but a third our size. And they were found in this complex cave in South Africa. It's really technically extraordinarily difficult because here's the entrance over here. Well, let me walk around here. Here's the entrance. Well, so far so good. All the way to down here. But the only people that could get through here and the chute right here were the skinniest of graduate students, all of the women. I can't imagine being in this hole down here and trying to climb up through here to get back out. Anyway, the bodies were all found here, and it was claimed by Berger and company several times that this was an example of reverential treatment of these bodies, meaning that they were certainly put in a protected place for sure, because no larger animal could get down there. But anyway, that's a controversy. But that's a third of a million years ago. Now here's something much more impressive. Here's an area west of Moscow, and here's the guess at what they might have looked like. Two children, one girl, 10, and a boy, 12, estimated ages, head to head. A lot of time was put there with various objects that were important. What's not shown in here were all the beads that were put in there, made from ivory, and hundreds of them, even thousands. That it would have taken a long time to prepare all of these beads, and quite an investment of the community to do that. So obviously these two children were very important to the community. We don't know why or how or what happened to them, but obviously the community committed a lot of time and resources to their burial. And that's around, 30,000 plus years ago. And that's the one we found. There are probably many more that we haven't found yet. Or maybe there's a high rise over top of it, or a highway, or an airport sitting on top of this stuff now. So I don't know how many more we'll find. Here's another fellow painted with ochre, red ochre, and some of the beads are still left around his head. So considerable investment has gone into this. And I've written something about this. Some of you have probably been to Egypt and visited some of these sites with very elaborate burial ceremonies, and they go back from almost Jesus' time, backwards 3,500 years from then. So 5,500 years from us, right? So a long time ago. And a very elaborate ceremony here. It's called the opening of the mouth ceremony. It took, if you were a person of some stature, it might take several months of taking organs out, processing the skin and kind of what's left, all the ceremonies that were attendant to this. So this is not you and I dying, having a service next week and in the hole. This is something going on a long time. And furthermore, many of the compounds that were used, the resins, came from the interior part of Africa or around the rim of the Mediterranean. So it tells you that there was an active trade going on. All right, so a very well-developed society. And of course this one, I was reminded of this by a former colleague here because her granddaughter is at the site right now, the Terracotta Warriors. Yeah. And, but the thing that was interesting to me about this, I can't, well, who was counting? But anyway, a lot of people were involved in this to create a city-sized mausoleum with these 700 terracotta warriors. The emperor didn't live all that long. He died at 49, possibly from toxic effects of potions to prolong his life. So maybe he got what he deserved, I don't know. Now we're going to switch gears here because that's a kind of macro picture to the cellular level, because that's really where the action is. And I think last week I mentioned that early in utero, in the embryo, many more cells of muscle and certainly the brain or the nervous system and other tissues are made but not retained. And there's an active competition between those cells in making connections. And the more robust ones survive and the other ones disappear without a trace. So that's what's called programmed cell death. Then beginning in the early 30s, remember that little depressing figure with the curves reaching their peak very early in life and then falling off. But that decline in numbers of nerve cells and connections begins in the early 30s, remembering that there's a lot of redundancy in the nervous system. You know, it doesn't mean that if 1% of your cells sell, you have lost 1% of your cognitive powers. It doesn't mean that at all. But it does mean the slope started. And the other thing is, we have stem cells. I don't know whether you know this, but of course it's obvious when you think about it. If you cut your skin, in a few days, with a Band-Aid on, it kind of glues itself together, and then the skin falls off 5 or 7 days later, or maybe 2 weeks later, and it all looks kind of pinky for a while, and then it kind of finally looks like a little kind of white, or a scar remaining. Well, where did those cells come from? They came from stem cells in the skin that are specific for skin. We have them in the bone marrow because we're constantly turning over red blood cells, white blood cells, any cell in the immune system. So we need stem cells to reproduce them. We know this is true with transfusions, right? Because when you're young and you're a donor, it doesn't take you long at all to make that up.
3 months.
There's an issue though, isn't there? Because with every reproductive cycle, there's a chance that the DNA will make a mistake, mean a mutation will be introduced. And that's fine because, first of all, the editing of mutations is very good. It's not perfect. And sometimes the mistake is big enough to lead to a malignancy. For example, I was talking to a friend in Niagara-on-the-Lake yesterday who told me that, he would be in his 60s, but that he had his physician found an abnormal protein level in his blood. And I know what that's called, it's benign monoclonal gammopathy. It's particularly common in men. And that can be benign for a decade or decades. But sometimes, at some point, it could turn malignant. And so that's what we're actually talking about. But that happens many times over. We probably have billions of nascent cancers that are nipped in the bud in our lifetime, and only a very few that actually cause us trouble, thanks to a vigilant immune system. We do have, you say, well, we literally live with the brain cells at much past the first year or two. We live with the brain cells that we were born with, except maybe for the hippocampus. And there is some evidence of neurogenesis, the creation of new brain cells in the hippocampus. But the problem is, it's in the nervous system, it's not just having a new cell, it has to talk to somebody. It has to receive messages and provide meaningful signals to other cells. If it doesn't make those connections, what's the point of having the cells? That's what's wrong with stem cells being implanted in the brain and somehow hoping against hope that they find some meaningful connections. Well, and that's the problem in the central nervous system. Hearing loss, this is a depressing thing, isn't it? Begins as early as the 20s. Well, I think we've marched through this. Just to point out at the bottom that stress itself, emotional stress, is that the area? Oh, okay. Emotional stress itself can, believe it or not, increase harmful mutations and lead to death of cells. That's something that people didn't appreciate for a long time. The cost of emotional stress, biologically. So, well, this is something. Until very recently, it was thought that DNA, there was a hierarchy in the cell, and DNA was the matriarch or the king of the cell, and it had the blueprints for everything else, and everything else followed according to what DNA said. That's not true. The cell is a cooperative. DNA is certainly very important, but so are mitochondria, so are other, the endoplasmic reticulum, all kinds of other cellular organelles and structures. They're all very important. And this is certainly true. We are born, it is not at birth, but the fertilized ovum, okay, starting with a single cell, and it divides, and it divides. About 20% of it becomes eventually the placenta, and the rest of it goes on through a highly orchestrated kind of routine to develop into all the hundreds and thousands of different cells and organs and systems, right? According to the code from the DNA. So we think, but there's more to it than that. Because what happens, beginning in the embryo, even very early in the game, is that there are other proteins in there that decide, after all, if one cell has the potential to become every other kind of cell, that can't work, right? You have to have a liver and a brain and special areas in your brain and special areas in your bone marrow. So that original totally potential cell with the potential to become any kind of cell has to, at some point, decide, or have it decided for it, your job is to become a liver cell. Your job is to become a whatever other kind of cell. How does that happen? There are proteins, methyl groups, that tagged onto a gene will silence it. And there is another, let's see where we are here. And so, well, here we are. So these methyl tags, I'm getting ahead of myself. These tags, these methyl tags, they increase as life goes on, especially during that period of development of the first third of life, and less so in the middle third of life, and not much after that. They define what that cell's progeny are going to become. All right? Together with another protein called a histone, which turns on genes. So it's a little orchestra. Think of it kind of biologically, there's a piano there, and it's playing certain keys in order to bring out a certain tune. And it's selecting the keys in the order in which they're played to become a specific type of liver cell, a specific cell. And you understand what I'm saying? So it isn't just DNA on its own dictating stuff. That's why identical twins aren't identical in their behavior and specific features, because their pattern of these epigenetic tags are different from one another. So they started out the same, but how it played out after that made them distinctive. So, here's a good illustration of it. There's Dmitry Belyaev, a Russian geneticist that Stalin would have hated if he'd known he was working. Probably would have killed him. But he was a scientist, and he was very interested in what would happen with, say, wild foxes that are inherently wary and aggressive. I mean, to get anywhere near them, they're trying to bite you. And they certainly don't want to do anything. I mean, put your hand in the cage and you're going to get it without a glove. So what he did is he mated in successive generations the most docile male and the most docile female through successive generations. And in no time at all, within 5 or 10 generations, these foxes became almost like pet dogs in their behavior, and didn't need a leash, and didn't bite. And the really interesting thing is that it wasn't just their behavior that changed, their appearance changed. Their snouts became shorter and rounder, the legs shorter and stouter, the ears floppier, and even the pattern and color of the fur changed. They became like domesticated dogs. So you say, oh gee, this must be an example of evolution on the fast track because of mutations. Not so. The genomes through these generations were virtually identical. It was the epigenetic. Okay, so here's the thing, no change in the genomic sequencing, but they identified 280 genes which were active in the prefrontal cortex that kind of damps down or controls emotions and that kind of thing, and wildness, if you like, that were active, but inactive in wild foxes. So same genes, but in one case turned off. Okay, that's what made them docile. And around the same time, I'll get to this in a second, there was a fellow Tremblay at the University of Montreal who was studying social factors and unwed mothers who were not very responsible about looking after their children, had a kind of rough life. They were rough on everyone around them, including themselves. And his curious observation was that it seemed to run into the next generation and the generation after that. How does that work? particularly when the second and third generations were, in many cases, had much better lives and much better provided for and a much healthier family support. Well, he presented this finding at Harvard, and it so happened in the audience were people who were very interested in epigenetics. And they said, can we study your patients in Montreal? And lo and behold, these mothers and their daughters and their granddaughters and great-granddaughters showed this epigenetic, this methylation pattern was almost identical. In other words, the epigenetic pattern had been passed on from generation to generation. That would have been heresy to say that 20 years ago in genetics, when all of the focus was on DNA. And people didn't appreciate these subtle but very, very important influences. And now histones, the other, so we have methyl groups and histones. They have opposite effects. So methyl groups silence genes, and histones excite them. And this is kind of a cartoon version of this. Remember a yo-yo? I remember as a kid playing a yo-yo. Well, histones looked like a yo-yo on a string, and the string around the yo-yo would be the DNA, right? Kind of unraveled from a chromosome and wound around the thing. And at certain sites along the way, different genes might have been silenced by attaching a methyl group to that strand of DNA. But at other points, there were these histones. Well, the histone is what do they call it? The
yo-yo.
Yeah, the yo-yo. Yo-yo itself has a little kind of string kind of hanging on it, and if you chemically modify that by acetylating it, you turn on the gene. So now we're beginning to realize, holy mackerel, this is already getting very interesting, and it explains a lot that classical. It's hard to think of classical genetics using even that word when much wasn't known until about the middle of the last century, right? Now we're moving on to another one. So any questions on that? All right. Just the point is, DNA is not, it certainly is a primary player, but it's not the sole player, and it's not the one that dictates which cells are going to become this or that, and how things change, okay? That's very important to realize that. Now here's an interesting observation. If you take a chromosome and the ends of a chromosome, the genes don't go right out to the end. There's a cap on the end of a sequence of bases, but not an encoding piece. So maybe 20 bases along that. So it's that little cap. As cells go through life and multiply and divide again, that cap starts to wear down. That's called a telomere at the end. It starts to wear down. There's no loss of any gene yet, but eventually, if that end wears down too much, then you start to lose active genes at the end. This phenomenon is called, after Leonard, Hayflick, the Hayflick phenomenon, meaning a cell can divide a limited number of times. And if it reaches that limit, because the cap's getting too small, that's the end of any further division. And the cells become what's been called senescent, aged, old. And it releases inflammatory factors, which can be destructive to surrounding tissues and cells. So the whole idea of the last 10 or 15 years, if we have these senescent cells hanging around and they're causing this mischief, why not get rid of them? Well, there's a whole field of senolytics, of finding drugs, pharmaceutical drugs, that kind of get rid of these senescent cells. It's kind of like getting rid of the garbage, if you like. Yeah.
These cells, they cause inflammation, and isn't it true that some inflammation then causes cancer? Is that why there's more cancer in people our age?
No, what happens is, we're going to get to that in just a second, with kind of the next thing, but it's a good question. Okay. So that's the limit, and it seems to hold true. Well, there's also, as you might expect, if there's a telomere, there's got to be an enzyme to make more of it. Okay. If you have too much of that enzyme making too much, adding too much to the telomere, it turns the cell into an immortal cell or a cancer cell. It can keep dividing and multiplying. There's no check in place. There's no time check, number of divisions before it stops. So senescence is a built-in check and on, well, not just in preventing cancer, but certainly that, for sure, plays a role. Now, one of the problems with all of this, I'm going to repeat this later, almost all of these studies have been done on mice. So I always had the writer, the observation was in mice, but very little of this has been confirmed in complex animals like us, or certainly humans at all. So what they say and what I'm saying here might not apply to us. And so what about house cleaning for these senescent cells? Well, I've even said hope for mice here. And this is the kind of overly enthusiastic observation. Removing senescent cells from mice made them healthier and delayed or even reversed age-related degenerative and inflammatory responses. Great for the mice. But mice live, what, a year and a half? So what are we actually talking about here? And is that even relevant to us? And where is the evidence in humans? Well, it's not very. So here's the science of senolytics. Is it real or hope? There is one interesting study in humans where they found that they developed a drug, this particular drug here, and in an experimental model of diabetic retinopathy, it seemed to improve the leakiness or reduce the leakiness and improve the visual damage in the retina of the mice. There is a study actually going on now in humans to see if the same holds true in humans. If it held true, that would be great. But don't, there's a problem with mice. It's not just because they're small, but they're genetically inbred. There's a lot of variation among us. There's very little variation among mice. And so what are you actually showing in a very genetically clean or specific species? And how does a year and a half actually equate to 80 in humans, or 90 years? It's hard to make that correlation. Well, now I'm jumping a little again, because this, believe it or not, they actually do come together. We're at 3 o'clock, so maybe we might not do this. And we'll leave this off to the next thing. So at this point, because I don't want to get into this subject if it takes another half hour or so, and I think it does. So we're going to leave this off to next week. I'm going to say a couple of things. This is a big subject. Hey, you know, I put on the series, I thought 4 would do it, but it actually is bigger than that. And you know, there's the whole subject of the influence of exercise and lifestyle on aging, which I haven't addressed and wasn't planning to address, but I actually think it should be addressed. So if you don't mind, I might add that on to another one to the series. It's no problem for me. I've got all the stuff, and it's not an issue. So if you're interested, we'll do that. But it's all very well to see all this stuff in Cells, and the literature's full of it, including the New York Times science section. But a lot of it's pretty soft science. This is not the kind of stuff that's getting any Nobel Prizes anytime soon. It might, but not so far. Yes?
Would you also add diet into the exercise as well, or that really would not affect aging?
I'm wary of getting to that because that's, if there's one overwhelming element of Fudge Factory, it's diet. Well, there are 2 issues. One is how much you eat and the quality of what you eat. And it's interesting, there are 2 books in my growing library on the subject, both written by one a cardiovascular surgeon and another by a cardiologist, but at least they share their heart. Both of them have written very charged books about lifestyle change and a lot of stuff on diet. But when I look at them, I think, first of all, it's a bad sign when they don't reference one another. That's a bad sign in science, because where are they getting the stuff from? And that's a no-no. And when I compared those 2, the only thing they had in common was the heart. And one of them was loaded with dietary advice, most of the foods I didn't even recognize. So I'm a little wary about that. But what is clear is that dietary restriction, I'm not talking about starving, but dietary limiting caloric input is that clearly works, especially if it's combined with exercise. Remember that I'm getting off subject here, I'm going on, rattling on because I'm on the subject now. It's a good time to bring it up. When you exercise, you go into the gym or you go for a long walk, and at a reasonable pace. I mean, I don't mean dawdling along, carrying your dog. I'm talking about really moving at a good pace. But if you do, the effect of that goes on for, I don't know whether you realize this, for about 35 hours, 35, 40 hours. The metabolic effect of that goes on for a long time. You continue, you see these little machines upstairs or down there on the, you know, all of those machines give you a little calorie count. What the hell's the point of that? When probably 2/3 or more of the calories are being burned after they leave the gym, over the next 24 hours or 36 hours. That's a gimmick. That's a gimmick to add that kind of thing. It does give you some relative sense of it. I mean, you do appreciate, as I do on the elliptical trainer, that if you kind of speed up, of course the calorie count's going to go up in me or consumption. And likewise for the brain. I don't know whether you realize this, but you know, the brain consumes 20 to 25% of the body's energy. You realize that? But the brain only accounts for less than 10% of the mass in the body. The brain burns energy like crazy. And just maintaining itself. And so hence its vulnerability and stroke or vascular disease, hypertension, hypotension. If the pulse stops, the clock is running. And although not as much, and then I'm going to finish right on this, stroke units around the world where, time is, or brain is time, or time is brain, that mantra has been around for what, 10 years or so? And it was started by a colleague of mine in London, Ontario. And it's true, but it's not as true as it hints. And why do I say that? Because I'm veering off here, maybe this will upset a few, but if you ask the question, how long does the brain actually survive? Well, we have very solid evidence on that because a very carefully constructed multi-center trial was done with a lot of people on the wings checking what was going on pigs. And so, the head's chopped off, preparation for meat and all that kind of stuff. Well, that brain is without blood. It takes 4 hours to reestablish the circulation there with an artificial blood. And they can dialyze the thing. But the point is 4 hours. If you look at that brain and you don't look at it, if you don't try to restore stuff, all kinds of bad things are happening, say by the 4 or 5 hour mark. But the amazing thing, if you reestablish the circulation at that 4 hour mark and doesn't come back to normal, but cells that were losing their capacity to control their water, that were their energy consumption going way down. They were on their way to death. They came back. They reestablished themselves. They were functioning normally. They were even talking to one another. And when they did electroencephalograms on these things, it looked like a fetal electroencephalogram, meaning parts of the brain were communicating with other parts of the brain. That was a stunner for the healthcare community, that study, and it's been extended to some other tissues because it has bearings for organ transplantation. And so the point is, I'm not suggesting rewiring me at the 4 hour mark. That would be a waste of time. But I am suggesting it isn't as tight as the stroke people would suggest. And that's why with successive stroke studies, surgical studies, and endovascular studies, that limit beyond which surgeons won't do it has slowly crept from 3 hours to 6 hours to 12 hours, and with reasonably good outcomes. So it clearly wasn't, a 3 hour limit, or minutes, as I was taught. What were you taught, Elizabeth? What were you taught in medical school? That the brain could tolerate a loss of circulation?
3 minutes was.
3 minutes.
But that's a long time ago.
Well, I know it's a long time ago. I'm as old as you are.
I'm very old.
Yeah, but I remember teaching that.
Yeah, we did too.
Yeah, and it was clearly was wrong. So next week we move on, and we'll add one at the end. Okay, is that all right?
May I ask a question?
Oh, are you? Oh,
Are you finished?
I am.
Okay. So, I previously have been taught that that we lose maybe 1,000 or 2,000 neurons a day,
Yeah.
you know? And so,
I'm counting mine.
well, this is, you know, every day of our lives that we lose so much. So I just want to know if you think that the brain productivity decline after 50, say, is related to a sort of an accumulated loss?
Like a threshold.
We're certainly losing cells for sure.
And then why do the neurons die? Some neurons die, those 1,000 die, and why do not others?
Well, one interesting.
What's the selectivity?
Yeah. Well, one of the we know what the oxygen consumption of the heart of the brain is, and it's very, very high. We used to think that we never paid any attention to the other two cells, thereby the glial cells or microglial cells. Turns out the glial cells, which provide an envelope around the neurons, are actually the source of lactate or glucose. And what happens in aging is that, it's a little slide further on here, but there's an enzyme that inhibits that. So in fact, the brain cells are starved for glucose. Well, the beauty of the study is that if you use a drug designed to block that enzyme, or you genetically re-engineer those glial cells so that it doesn't happen, the brain cells remain quite healthy. So, but you know, these studies are done on relatively simple animals. I mentioned mice, rats, that kind of thing. They're cheap. They have a short lifespan. You can do a study in a short time. You know, aging is a matter of weeks and maybe a few months. It's not 40 years. And but those studies are misleading. And so when I look at the literature, I see little light bulbs going off in centers who are really interested in mitochondria or are really interested in senescent cells. And they're really enthusiastic and energetic, and they've got a whole lab working on that, but they're not looking anywhere else outside of their own interest. And the problem is that doesn't really translate often into meaningful stuff for you and I, meaning a medication that would mitigate all of the things going wrong. You see, the issue is that people say, oh, well, what happens with aging happens because of mitochondria, happens because of mutations in DNA, it happens because of the garbage system isn't working. As far as I can tell, looking at the literature, aging is affecting every system within cells. There isn't a godfather system that goes and all the rest fall in line. They're all kind of wearing out. It's like, I don't know what your car is like, but you know, all cars don't die because the transmission goes or the ignition goes. They die because the tires wear out, certain gears go, things happen in the engines, not at the same time, but if you have a car for a long enough time, you'll find out about all these other systems. And cells are like that too. All of these things have a life period. It happens, I know because I own several airplanes, stuff happens. Stuff is life or time limited. So an engine would have a TBO of 2,000 hours, and you would have to take it in and recondition it or get a new one. That's it, 2,000 hours. The early jets, the German 262, had a, believe it or not, a life of 10 hours. And why was that? Because they couldn't lay their hands on certain rare metals that would have made it tougher and more resistant to heat. So anyway, we're well beyond stuff.